I am Germaine WAKEM, PhD student in University of Yaoundé I, under the direction of Professor Emmanuel Yumbi of the University of Yaoundé I, Professor Robert Joinke of the University of Ngaoundéré and Dr. Behaviour from CIRAD. It's my pleasure to present to you our work under the title An Attempt to Differentiate Cassava Genotypes by their Retting Behavior using some Biophysical Indicators. Cassava is an important food resource and it's now the second most important commodity after corn and occupies more than 50% of total RTB production. Despite its importance, the harvested cassava root faces two main difficulties. Firstly, the perishability of the root. In fact, the harvested cassava root can only be stored for a period of 1 to 3 days. After this period, they undergo physiological deterioration, which damages the quality of the root. Secondly, some varieties contain high levels of cyanogenic glucosities, which are very toxic substances and involved in serious neurological disorders, which can lead to death. Faced with these problems, the actors react on the one hand by developing transformation processes in order to convert cassava roots into various more stable products. One of the best known practices on the peasant scales that will hold our attention in this work is wetting. Wetting is a conventional process that involves soaking cassava roots in water for a period of 3 to 7 days. The purpose of wetting is to soften the root in order to facilitate their malleability in derivative products. What's more, microorganisms and enzymes degrade cyanogenic compounds and improve detoxification of derived products. On the other hand, research actors have developed new varieties. However, despite their higher yield than local varieties, their adoption by users remain limited because they do not contain the same quality properties. This contributes after some time to the abandonment of these new varieties by the peasants. As the rating ability being one of the major criteria which condition the adoption of new varieties, the objective of this work is to identify the biophysical indicators associated with the cassava rating ability of some cassava genotype. The plant material was collected in experimental plot set up by International Institute of Agriculture. This material consisted of 8 varieties of cassava, including 7 improved varieties, including 4 with yellow pulp, 3 with white pulp, and local variety with white pulp. The experimental model being a comparative biochemistry approach on a contrasting genetic model. Approximately 30 kilograms of root of each harvested varieties were peeled, washed, and divided into 3 batches of approximately 10 kilograms each and soaked in tap water. The rating test lasted 3 days, in which the roots were randomly sampled from each batch and stored in formaldehyde for microscopic examination on the one hand and frozen on the other hand. The frozen samples were then freeze-dried and ground. The resulting flour were used for the amylose, starch, and starch fasting property analysis. A part of the material was transported to Montpellier, where a rating test was carried out and the firmness directly measured in pieces of root taken at random every 24 hours using a texturometer. This method allows us to obtain several results, the most salient of which are as follows. This figure is the graphic representation of the change in firmness during rating, with the firmness on the ordinate and the duration of rating on the abscissa. Each color is in the histogram represents the variety. In general, the loss of firmness is effective in all varieties, and after 48 hours, the rating is almost complete. but the varietal contrast was very pronounced after 24 hours of rating, with the varieties 01, 00, 40, 27, and LMR, which almost completely lost their fewness. The varieties 92, 03, 26, 01, 1797, IO 7, 10, 26, IO 7, 05, 93, and IO 9, 06, 16, which are intermediate, and the local variety which has remained relatively thin. On the basis of this firmness criterion, which moreover represents the main characteristic criterion of rating, we have chosen of the following biochemical analysis: 2 varieties with rapid rating and 2 which are relatively slower. This figure shows the histological section of the fresh roots on the left and after 72 hours of rating on the right. On these images, XC represents xylem self, CW represents cell walls, and SG represents starch granule. Each letter represents a variety. At harvest, the variety exhibited a consistent structure with cells containing the granule starch and with a turgid appearance, except the local variety, which exhibited plasmolized cells. The cells were larger in an LMR 92, 03, 26, and 01, 1797 varieties. After 72 hours of rating, the results show a degradation of the cell structure without apparent modification of the starch granules. This degradation was more marked in the variety IO 7, 10, 26, IO 40, 27, and the local variety, while the variety IO 9, 06, 16, and RMR represented a still cohesive cell structure with relatively continuous cell walls. Although rating was effective in all varieties, microscopic examination showed a marked varietal contrast in cell structure. Therefore, we can say that rating destroyed the root structure by completely degrading the cell walls in some varieties, while the starch granules do not seem to change during rating. These histograms show the amylose and starch contents of the roots at harvest, with the varieties on the abscissa and the amylose and starch contents respectively on the ordinate. It appeared that the variety 01, 1797 had the highest amylose content, while the LMR had the highest starch content. Therefore, the varieties with comparable starch content had different amylose content, which also suggests different amylopectin levels in these varieties. Although this amylose content could be considered as a discriminating factor between varieties, overall, all these varieties are considered to be low amylose varieties because they contain an amylose rate of less than 20%, which makes them less susceptible to retrogradation. This is a graphical representation of the evolution of peak viscosity during rating, with the duration of rating on the abscissa and the viscosity on the ordinate. Each color represents a variety. Peak viscosity is the ability of starch granules to absorb water without bursting during heating. On graph A, only 01, 1797 variety show an increasing peak viscosity of 3.92% compared to untreated roots. So, the rating improved the swelling of the starch granules in this variety, unlike the other varieties. At T0, the 920326 variety exhibited the highest peak viscosity, which means a high water retention capacity of the starch granules and therefore a high swelling, consequently a higher degree of gelatinization compared to other varieties. Although this variety was not the one with the lowest amylose content, it did have the highest peak viscosity. However, it was shown that a low peak viscosity is a consequence of the high amylose content. This is valid in the case of the 1797 variety, which has a low peak viscosity in response to its high amylose content compared to the others. But this difference in gelatinization between varieties is felt only in unrated rows. We have on these histograms the graphical representations of the evolution of the breakdown and the setback viscosity during rating, with on the abscissa the duration of the rating and on the ordinate the breakdown and setback respectively. Each color represents a variety. The breakdown expresses the resistance potential of the granules to heating, while the setback reflects its susceptibility to retrogradation. These two parameters decreased in variety 920326 and 1797 by 27.77% and 22% for the breakdown, and 13.63 and 30.41% for the setback respectively during rating. This means that rating will increase the resistance to heating of the granules while reducing their susceptibility to retrogradation in these varieties. This variation in the breakdown between varieties reflects the difference in the stability of the starch granules when heated. Variety 920326 exhibited the highest breakdown and setback at harvest, which means it's less resistant to prolonged heating and exhibits a higher susceptibility to retrogradation compared to other varieties. This PCAB plot carried out on variable and varieties after 24 hours of rating mobilized 84.6% of the total inertia. Variety 920326 and 1797 seem to be characterized mainly by high peak viscosity, setback, breakdown, and firmness, while variety LMR and 004027 seem to be characterized only by peak viscosity for one and the setback for the other respectively. But no significant correlation was found between the parameter study. It's believed that other factors such as the size of granules, the length and distribution of the amylose and amylopectin chains, and degree of polymerization will influence the fasting property. The dispersion of cassava varieties in the first two dimensions reveals the influence of genotype in the rating process. In conclusion, we examined the biophysical indicators during the rating of 8 cassava varieties. It emerged that all the varieties studied have a low amylose content. The rating seems to affect the cell walls more, while the starch granules only seem to be remain unscattered. According to the PCA, there is no correlation between fasting property parameters and fitness. The hydrothermal behavior seems to be driven by a differentiation of the structural organization of the starch granules. In perspective, an analysis of the constituent of the cell walls would make it possible to accurately determine those most affected by rating. Other biochemical compounds, such as phenolic acid, would be analyzed in order to determine their implication on the rating capacity. A starch crystallography would allow a better understanding of the structural organization of the granules of each variety in order to better understand their behavior during rating. We cannot end without expressing our gratitude to our partner and founders. Thanks for your attention.
Thank you very much, Germaine, for this very interesting and promising presentation. I think it's one of the first results that we see on a possible link between the cell walls and modifications to the cell walls during the retting process and the fairness or texture of the final product. I think we have about 10 minutes for questions. I would like to invite any questions from the audience. Okay, I had a question on the harvesting protocol. You mentioned that you had that 30 kilograms of roots were harvested randomly. Can you tell us a bit more, like how many plants did you have in the plot in total, and how many plants did you harvest to get 30 kilos?
Est-ce que je peux répondre en français?
Oui, on pourra traduire.
Nous avons récolté en moyenne 10 plants par variété. Mais avec, pour toutes les variétés, avec les 10 plants, on n'atteignait pas les 30 kilos. Donc, pour certaines variétés, on allait jusqu'à 12 plants. Mais en moyenne, c'était 10 plants par variété.
D'accord. So the harvest on average, 10 plants were harvested per genotype. And in some cases with 10 plants, there were less than 30 kilograms. So in that case, up to 12 plants were harvested. We have a question from Miriam.
Can I add, Thierry?
Yes, please, Didier.
I would like just to add a precision. The plant were harvested randomly on the plot. The 10 or 12 plants were randomly harvested on the plot.
Okay. But because in the plot you may have plants of like strong, healthy plants and plants that are a bit weaker. So there was absolutely no selection to take the more healthier plants?
No, there were no selection.
Okay.
We harvest with IATA detection.
Yeah, because from experience, I know it can be tempting to, if a plant looks a bit weak, just to leave it on the side and to take only the better plants.
Yes, and what we did, we harvest randomly between 10 to 12 plants. We collect all the roots, and we selected the roots based on their size.
Okay, yeah. Okay, so it was good.
To reach 30 kilograms.
Okay.
Varieties.
So there was some selection of the better roots.
Exactly.
Okay. Thank you. We have a question of Miriam Ofose. Miriam, would you like to ask your question, or shall I read it? So the question is, what was the surface area of the cassava roots put into consideration during retting? Donc Germaine, est-ce que la surface de contact, la surface de la racine de manioc pendant la fermentation, pendant le rouissage, a été prise en considération?
C'est vrai que je ne saisis pas très bien ce qu'elle veut dire par surface de la racine.
Meaning that if you have smaller roots, you would have a larger surface and then possibly a faster retting because there is more surface to be attacked and to be fermented by the bacteria. So, si on a des racines plus petites, on peut avoir une surface d'échange plus grande et donc avoir un rouissage plus rapide. Est-ce que les racines étaient coupées en morceaux?
Oui, nous avons découpé les racines en morceaux. Mais la taille n'influençait pas en fonction de la variété. Donc, il y avait les variétés qui ruissaient normalement sur toute la surface de la racine, indépendamment de la taille du morceau.
So the roots, the answer is the roots were cut in pieces of similar sizes. So normally the surface of contact did not influence the retting process. Question from Emmanuel. Germaine, did you take the age of the cassava roots into consideration? What was the age of the roots at the harvest? Donc, quel était l'âge des racines au moment de la récolte?
Les racines avaient 12 mois. 12 mois. Oui, 12 mois.
Pour tous les génotypes.
Oui, 12 mois pour tous les génotypes.
Okay, so all the roots were 12 years old at the harvest for all the genotypes. And what was the season of the harvest, or what months did you do the harvest? À quelle saison avez-vous fait la récolte?
Nous avons récolté en juin.
Okay.
Le mois de juin.
Ok. En juin de 2020, du coup. De cette année?
Non, en juin 2018.
2018, ok.
Oui.
Okay, so the season for harvest was in June. I wanted to ask you as well if you were able to extract some quantitative parameters, or if you're trying to extract quantitative parameters from the microscope photos to try to analyze more in details the degradation of the cell walls. J'avais une autre question, c'est savoir si vous avez essayé d'extraire un paramètre quantitatif pour caractériser la dégradation des parois cellulaires sur les photos au microscope qu'on a vues.
Non, pas encore. Nous avons mis dans les perspectives qu'on devrait quantifier les constituants des parois cellulaires pour voir quels sont les constituants les plus touchés au cours du ruissage.
So the answer is not yet, and one of the work being planned is to analyze which components of the cell walls are most affected during the retting.
Can I please complete the answer?
Yeah.
We also
please,
plan to treat the picture, the microscopic picture, and try to quantify the star granule or the density of the cell based on the picture. But it was quite difficult for two reasons. The first one is that we failed to control the soft. And the second reason is that according to us, it was very difficult to do that because from each variety to one, or from each sample to another, the picture was not taken at the same place, at the same area. So it will be very difficult for us to compare the variety each other or the picture each other. That's why we didn't go in deep in this way.
Okay. Thanks, Didier. We have two questions from Ernest Chukudi. So, sorry if I mispronounce names. So what was the percentage yield of the mash in relation with the root weights? So quel était le rendement de la racine broyée par rapport à la racine fraîche? Peut-être avant et après bruissage.
Nous n'avons pas évalué ces paramètres. Donc, quand nous avons fait le ruissage, ce qui nous intéressait, c'était les paramètres physico-chimiques qu'on devait prélever sur place. Et la pâte fermentée, on n'a pas eu à mesurer quoi que ce soit dessus, même pas le poids, rien.
So
Je ne sais pas si ça répond à la question.
Oui, je vais traduire. Okay, so the answer is that the yield was not a focus of the study, so it was not evaluated in this series of experiments. And then the next question is, were there some roots that did not rate very well? Donc, Germaine, est-ce que certaines racines ou certains génotypes n'ont pas fermenté ou ne se sont pas ramollis aussi bien que d'autres?
C'est vrai que la cinétique du ruissage était différente d'une variété à l'autre. Mais d'une manière générale, à la fin du ruissage, toutes les racines avaient rouillé. Et concernant la question de pourquoi, quels seraient les facteurs responsables, je pense que c'est ça l'objectif de notre travail. De trouver la raison pour laquelle certaines variétés ruissent plus vite par rapport à d'autres. Peut-être que, bon, dans notre cas, toutes nos variétés ont rouillé. Donc, il est question pour nous de savoir, de trouver les paramètres physico-chimiques qui expliquent le pourquoi de la différence dans cette cinétique de ruissage.
Okay, so I will translate. So Germaine answers that yes, the kinetics of retting were different for different genotypes. Some genotypes become softer, faster than others. Nevertheless, after 48 hours, all the genotypes in the study were soft and completed the retting process. So the key objective of our study is to identify the factors responsible for those differences in retting kinetics. And this is still ongoing work. They don't have definite answers yet. But we may suspect that the cell walls are involved, because in the end, the differences in terms of starch, amylose, were not that significant or not that strong. We have time maybe for just one more question. Anyone from the floor? If not, I would like to thank Germaine for her presentation, as well as the team of our supervisor. And thank you to everyone for your time in joining us today.